Okay, so we talked about, once again, we see that DNA, the change there. The T and the A has been changed, right? The T and the A has been changed. And we also look at the codons. So we said that CTC, remember that that is DNA. That's the DNA right here. This strand is DNA. So when we go to the messenger RNA, we're going to be reading GAG. And we said that GAG is glutamate, right? Now we look down at the bottom one. We're reading CAC, which translates into transcribes into GUG, which gives us valine. So we have changed the amino acid, right? It's still only one amino acid that's being changed. Does that make a difference? Absolutely. Okay, absolutely. Now, looking at this, up at the top, that is a normal red blood cell. Okay, so this is part of the code for a normal red blood cell. Okay, have you ever heard of the sickle cell? Have you ever heard of sickle cell anemia? Right here is the sickled cell. So, by changing that one letter from going from that T to that A, one letter, okay, changes from a normal blood cell to a sickled red blood cell. All right? So does it matter? Yeah. Okay? Yeah, it does. Now, that is what we call a mutation. A mutation is any change in the DNA sequence. Any change. Now, when we talk about mutations, okay, if I would ask you about the word mutation, what would you tell me? It means change, okay? Means change. If I would say that I got a mutation, would you think that would be a good thing? Okay? We typically think of a mutation as being something that's not good, right? But is that always the case? Right, we could have changes that would help us, right? We could have a change that could help us. This usually happens during DNA replication. Okay? It usually happens during DNA replication. Remember, during DNA replication, the DNA polymerase is bringing the bases one at a time, right? One at a time. Do you think that that polymerase could possibly maybe read something wrong? Bring in a wrong base? That could happen. It could happen. You would be surprised at how efficient our bodies are at DNA replication. Okay, very rarely are mistakes made, but there could be mistakes. All right. So if we look here, here's just another instance of a error. Okay, we're looking at this base pair right here. Okay, I look at this base pair right here. When I have the original copy, I have a green with a purple. Now, on this copy, I have a green with a red. So I have definitely changed that DNA, haven't I? That would be a mutation. Could occur during replication. If the mutation is in a sex cell, it may affect the offspring. the children, the next generation. Okay? So if there was a mutation, if a father had a mutation in his sex cell, he could possibly pass that on to his kids. However, if the mutation was in a body cell, just a regular body cell, not the sex cell, if it was in a body cell, the only one that would be affected would be the individual. Okay? So in order to pass on to your children, those mutations need to be in the sex cell. Okay? Mutations can be bad. We talked about that. We can have mutations that lead to cancer. Could cause you to age faster. Could lead to birth defects. We could also have mutations that would cause embryos to self-abort. Some sort, maybe a miscarry. Okay? So mutations can be bad. Those are all things that I wouldn't consider very good. Okay? If you look, here's a little chick, or a little duck, I guess. It's not a chick brawn. It's a duck. Okay, I'm getting better at my animals. Okay, that one's a duck. Okay, looks like that duck has a little extra foot there, doesn't it? Right there. Okay, there's a foot. There's a foot. What's that guy? Okay, right there's a mutation. Okay, there's a mutation, and that looks to me like a cat with no fur. Huh? Okay, I'm not real specific, or don't know the specific name of it. I'm not real big on cats. Okay, doesn't look very good to me though. Okay, here's a dog. I like dogs better. Okay, I can't really tell, but I would say probably what that is maybe he didn't have another leg. I don't see a fourth leg. Do you guys? Now, it could be hiding, but I wouldn't understand why they would put those wheels on there if he had. I would say probably the mutation was that he was born with 3 legs instead of 4. Okay, so they put that little scooter on there to help him walk. Mutations can also be good. Okay, making an organism survive better in its environment. That would be a good thing, right? Now, an example is bacteria that becomes antibiotic resistant. You know, antibiotics, you ever heard of them? What's an antibiotic? Dylan? It's a medicine. You might get it from your doctor. Let's say you're sick, you go to the doctor, he says, I'm going to write you a prescription for an antibiotic. Okay? So, with this antibiotic, that bacteria that's making you sick, it kills that bacteria. Now, what if the bacteria wasn't affected by that antibiotic? Wouldn't that be a good thing for the bacteria? Okay? Yeah, it would be. So if you look at this, what these are, each one of these dots are antibiotics right there. Okay, you see the yellow part here? This yellow part is bacteria. We've grown the bacteria on the Petri dish. We put the antibiotic on there. What happens to the bacteria? See those circles? It's killing that bacteria. Look at this one, though. Is that bacteria around that circle being killed? No. So for some reason, bacteria there, look at there. Okay? It's a little dimmer, but there's still more bacteria living there than, let's say, over here, right? So some of that bacteria has become resistant. So it has mutated so that the antibiotic does not hurt it. Okay? Here it says the ability to drink milk as an adult is a helpful mutation. You've probably heard of people that are lactose intolerant. Okay, your stepdad is. Okay, lactose intolerant. So they don't want to drink milk, right? It'll make them sick. Okay, it'll make them sick. Also, mutations can have no effect. Okay, it could be good, could be bad, could be nothing. What do you mean by that? A mutation doesn't have any effect? Well, let's look at this. We have a CAC. We were just talking about that, right? That translates into what? Valine, correct? Look at CAT. Okay? So we're going to look at GUA, right? What's GUA? What is GUA? It's also valine, right? So did we change? Did we have a mutation? Yeah, we changed from C to T. But it still read for the same amino acid, didn't it? So are the amino acids there changed? No. Would the protein be changed? No. So was there a mutation? Yes, there was. However, it's still coded for the same amino acid. Remember, we only have 20 amino acids, right? Look at your codon chart. There's more than 20 codons, aren't there? So some of those amino acids are repeated. That repetition is what happens when we have this mutation and there's no effect. Okay? Now let's talk about the types of mutations. Look at this one right here. Can we see what has happened in this mutation? There's no D, is there? Okay, so I have A, B, C, D, E, F, G, H. However, on my right-hand one, that D is gone. We call that a deletion. We call that a deletion. It changes the number of bases by removing a piece of DNA. If we remove that little D chunk right there, do you think it's going to affect the amino acid, the protein? Absolutely. Now, here is an example right here. See the CAT? We took out the A. So we have deleted that A. Now, instead of, if you look, all of these are HIS, HIS, HIS. Now, if we took out that A, we moved everything down. So now instead of CAT, my codon is CTC. Then I have ATC and start repeating a lot of ATCs. So the amino acid sequence is different because I removed that one base. Look at this one. What's happened there? What's happened there? The ABCD is now ADCB. So it's flopped. We call that inversion. Inversion. The entire section of DNA has been reversed. The entire section of DNA is reversed. It's inverted. Now, if I show you this slide, look at this. Okay? What's happened here? What's happened here? Can you see that this little part right here is now up top? Does that look the same? No. We've actually just taken this right here and flipped it. The blue parts are still on the bottom, but we've flipped where that centromere is, haven't we? Look at this one. Okay? What's happened here? I have 2 genes. What's happened here? What's happened here? Can you tell? You see anything different? They flipped, right? The AB went to the bottom one, MNO went to the top one. That's something like crossing over, right? So that is a translocation. A translocation, a whole chromosome or a chromosome segment attaches to another chromosome or another segment, thus creating a hybrid. We talked about crossing over before and how that helps with the genetic diversity. Okay? That is what's happening in this translocation. The DNA is crossed over to a different chromosome. Now, if you look at this, okay, here's chromosome 4. Here's chromosome 20. Now, look what's happened. I've taken this little box of 20 and put it on 4, and taken this big box on 4 and put it on 20. So, the segments there of the DNA have switched to new chromosomes. Do you think it makes a difference? Absolutely. Translocation. Here's another type. Okay? Here's another type. What's happened here? What's happened here? We've added a C, haven't we? We added this C right in there. Okay? And we'll talk about what this means here, this frame shift. But we've added a C. That is insertion. It increases the number of DNA bases in a gene by adding a piece. This one, we just added 1 gene. Could we add a bunch of genes? Okay. Could it be more than 1? Absolutely. In this case, it is only 1 right here. So we've just inserted that 1. Now, a frame shift. A frame shift, I told you we would get to this. A frame shift shifts the grouping of the bases and changes the codon for amino acids. Can you see on here that now this codon was C, T, G. Now we've got C, C, T, and we've shifted every other one down more and more and more. Okay, a frame shift shifts the grouping of the bases and changes the codons for the amino acids. If you look at this, okay, can you see that insertion there? I've taken this box of chromosome 4 and put it into chromosome 20. So now chromosome 4 is much shorter. Chromosome 20 is longer. So I've inserted a little chunk or a little segment of that DNA. Here's our last type. What's happened here? It went A, B, C, D, E, and now it goes A, B, C, B, C. So we've inserted another BC here. So basically, we've duplicated this BC, and now I have this BC twice. That is what we call a duplication. A piece of DNA that is copied 1 or more times. So it could be A, B, C, B, C, or could be A, B, C, B, C, B, C, B, C, B, C, B, then go D, E, F, G, H. Okay, so it could copy multiple times, not just that one. Now, if you look here, I have a section of DNA right here on this chromosome. It's in yellow. Okay, it's in yellow right here. I have that same one, but now it's duplicated again. So I have it twice. So the duplication of that piece of DNA, that chunk, that segment, has happened twice. Now we've talked about these mutations. What causes them? What causes a mutation? Well, it can be inherited. It can be environmental, or it can just happen spontaneously. One of those three things. Can be inherited, can be environmental, or it can happen spontaneously. A mutagen is anything environmental that can cause a change in the DNA. So, if something is a mutagen, it causes a change in the DNA. What are some mutagens? Radiation, ultraviolet rays from the sunlight, X-rays, okay, nuclear energy. Those all can cause mutagens. Okay. Chemicals. Okay, chemicals are mutagens. Asbestos, formaldehyde, chemicals that are in tobacco products. All of those are mutagens. Many of these mutagens are what we call carcinogens. I'm sure we've probably heard of a carcinogen, haven't we? A carcinogen is just something that is cancer-causing. Okay? Could someone, let's use this example. Could someone get lung cancer from smoking cigarettes? Right? Absolutely. That is a carcinogen. Okay, let's go back and think about radiation. Okay, could somebody get skin cancer from being in the sun too long? Yeah. Once again, that ultraviolet ray could be the carcinogen, cancer causing. Okay,